All right, so this will be our last lecture for this class, and we're going to focus in on gene therapy, specifically genome editing, and we'll even talk about cloning a little bit at the end of this as a mechanism for gene therapy. All right, so the first question we need to address is this one. What genes are candidates for gene therapy? What do we need to know about a gene in order to implement gene therapy changes? So we have to know several features. First, we need to know which gene is involved in the disorder, and this seems like a no-brainer. But we really only understand a very small part of the human genome still. There are several genes that we don't know what they do, and there are several genetic illnesses or even genetic conditions that are linked to particular genes but aren't caused by those genes. So this can become pretty complicated to try to figure out what genes are actually responsible. Plus, if you have to do gene therapy using multiple genes, the whole procedure becomes far more complex. So, in an ideal case, like let's say cystic fibrosis, we know the gene that causes the disorder, and so this would be a good candidate for gene therapy. We need to know what a normal copy of the gene looks like. Again, using cystic fibrosis as an example, we know what a normal copy of the gene looks like because we've studied this gene in great detail. We know what the protein product looks like, we know what it does, we know how it should operate. We need to know when and where the gene is expressed. We need to know in what tissues does the gene get expressed? And we also need to know from that when the gene gets expressed. Does it get expressed during development? Does it get expressed all the way throughout the life of the organism? Does it get expressed, after the cells go through the cell cycle, go through a mitotic sequence? And then finally, we need to know what happens, um, what happened expressed normally. Wow. That should say what happens when expressed normally. What does the cell look like? What protein products are formed? How does the cell behave? How does the cell work mechanically when the gene is expressed normally? And all of these, show that cystic fibrosis is a great candidate for gene therapy. And we've talked about this before with genetic screening for cystic fibrosis and some other mechanisms. So there are different ways in which you can do gene therapy. Today we're going to focus in on CRISPR. We can talk about a number of different techniques, but CRISPR seems to be, at least right now, the most fruitful of all of our techniques for genome editing and gene therapy. All right. So the next question is this one: what must gene therapy do? What must gene therapy do? So not necessarily okay. So now we've got past the point where we have a gene, we know what it does, we know where it's expressed, when it's expressed, we know what a normal copy of the gene looks like, and we know what the cell does when the gene's expressed. But how do we then implement gene therapy? What does gene therapy need to accomplish? Well, first we need to be able to get the genes in the proper cells. We need to know where the gene is expressed and when it's expressed, and we need to be able to get our gene, the correct copy of that gene, into those target cells. And then we need to make sure that those genes are expressed. It's not just getting the gene in there, but you have to also ensure that that gene gets expressed in those target cells. And not just that, but you need to get that gene to integrate into the genome of the target cells so that when these target cells go through mitosis, they produce daughter cells with that particular gene in it. And this, of all of them, this is one of the more challenging. And this is one of the reasons why that gene therapy tends to be short-term success in humans so far, is that it's incredibly difficult to actually integrate this gene into the genome. And this is one of the reasons why we're going to focus on CRISPR, and because it provides us one of the best ways to incorporate the gene exactly where we want it in the genome. And then also helping us, we need to be able to avoid side effects, unintended consequences. And this is another very complicated one. We know over 90% of the genome is actively transcribed. And so you can't just add these genes anywhere in the genome. You have to add them in very specific places and know exactly what you're doing when you add them into there and disrupt what's already present in the genome. And this is, again, one of the reasons why CRISPR is so exciting, because we can target exactly where we want our gene to insert into the genome. The next question: how do we actually do gene therapy? And so there are three strategies, or three categories. Strategies is probably a good term of gene therapy. The first one is gene augmentation. This is often used in genetic conditions that have a recessive inheritance. So you have somebody somebody that's a double recessive. They are expressing whatever that phenotype is. They have that genetic illness. What we can do is add normal copies of that gene, wild type copies of that gene, to make sure that the proper protein product is made and that they express the dominant phenotype, which would be the lack of that genetic illness. The next two tend to be ways in which we combat cancers and other genetic illnesses. So one of them is targeted cell killing. In here, we put a gene in that is either going to make a toxic product, or we put a gene in that is going to cause the cell to express a foreign cell receptor, either to signal to the immune system that it is not self, even though it is self. This we oftentimes use as gene therapies in cancer. So this is also known as suicide gene therapy, because the cell that is expressing whatever this gene is ultimately going to die as a result of that gene expression. The last one is targeted mutation correction, and this is where you have a genetic illness that has a dominant inheritance. So all you need is one version of that allele in order to have whatever that genetic illness is. And so you actually you can't just put in the proper form of that gene, because whatever this is expressing and masking the wild type. And so this is true of a lot of cancers as well, with a lot of oncogenes, where there's a mutation in an oncogene that may, say, distort the cell cycle, or leading to uncontrolled cell growth, or the inability of the cell to go through apoptosis. And so what you have to do in this type of gene therapy is you actually have to target your mutant gene, and you need to destroy it. And sometimes that's enough to just destroy that gene and prevent it from being transcribed. But you could at the same time replace it with another version, a different allele of that gene. And so this is where CRISPR actually provides, again, some really exciting opportunities and thoughts on how we can do this. So let's focus on CRISPR. Let's talk a little bit about CRISPR, and I know we had a group earlier in the semester. They presented on CRISPR, and specifically CRISPR-Cas9, and talked about it in really good detail and explained it well. But I want to review that and then talk about the potential of this with regards to treating genetic illnesses. So CRISPR is not the only strategy for genome editing. That should say genome editing. Man, second typo. It is not the only strategy for genome editing. There are several where you can go in and you can cut the genome in specific places using restriction enzymes, and then replace or insert a new gene. Or you can use viruses. That's what historically gene therapy has used primarily, is viruses to insert specific genes into the genome. But CRISPR-Cas9 is the easiest, and it's the least expensive, and in many ways some of the most targeted as well. And so it's again, like I mentioned earlier, it right now seems to be the most productive and the strategy of editing the genome with the most potential. And so, again, like the group mentioned earlier this semester, it was adapted from bacterial immune systems. that basically make RNA strands to target viral genomes. And so these RNA strands basically are made as complements to the viral genome. And so when the virus, any future infections with that virus, those RNAs are going to bind to the viral genome, which prevents the viral genome from incorporating into the bacterial chromosome. But it also, on top of that, these RNAs signal Cas9, which is a restriction enzyme, to come and to cut up that viral genome. And so the way CRISPR works, again, in bacteria, they make RNA complements to that viral genome, but we can actually design specific RNA molecules that will bind to whatever region in the DNA we want them to. And so we'll make modified RNA molecules to bind to, say, a section in the genome that has a mutant allele that has a dominant inheritance. So one copy of the mutant allele will lead to the particular genetic illness. So we can design an RNA to bind right there in the genome. And like in bacterial immune systems, it will recruit Cas9, and then this enzyme will cut the DNA at that location where that modified RNA is bound. And the beauty of this is it doesn't just cut the strand of DNA that the RNA is bound to, but it actually cuts both strands of the DNA. The strand that the RNA, the modified RNA, is bound to, but also cuts the complementary strand to that as well, producing what's called a double-stranded DNA cut. And those are relatively complicated to repair, which is nice. The cell can repair those, but it's complicated. And so what that provides is a mechanism by which you can insert a specially designed version of the gene. So the DNA repair machinery of the cell, or better yet, a custom DNA segment, can actually be put into the cell, and there's a pretty high probability that this custom DNA segment, if it matches up at that double strand break, that you're going to get that to incorporate in and forming recombinant DNA. And so you can target this, you can target the genome at that specific allele. You could cut it, which is going to destroy that allele, prevent it from being transcribed properly and then translated into the proper protein. And where it's cut, actually insert the normal version of that gene, the wild type allele. And so as that gets transcribed, it will get translated in the correct way. And so here's an overview of Cas9. Here's this modified, the specially designed RNA that will go in and will bind at your target region and will recruit Cas9, which again is that enzyme, and then it will cut this at a very specific place. And then what you can do is you've now cut this, produces a double strand break. And again, this is relatively difficult for the cell to repair. And so you can design a piece of DNA, which, ideally is the normal allele for that protein. And there's a pretty good chance that you're going to form recombinant DNA here. You're going to insert that replacement gene in. And so it's incorporated in the genome. So as far as what you need to do with gene therapy is you found your target cell, you got this gene incorporated into the genome, and it will be expressed. And ideally, you've inserted it in the exact place where you destroyed the bad allele, and you've replaced it with the proper allele. And so it should in many ways avoid some of those unintended consequences that you're worried about with gene therapy. And again, this is all ideal. We still have a long way to go in figuring out how all this is going to work and what it's going to mean. And again, for many genetic conditions, it's not a single gene. It's a suite of genes, and it's far more complex. But this is really providing a very exciting and a potentially very fruitful area of research in genome modification and in gene therapy to treat particular genetic illnesses. And then lastly, we need to talk a little bit about cloning. And so cloning isn't directly a gene therapy, but can certainly be used in gene therapy. And we'll talk about that as we talk through the details of cloning. So first of all, for cloning to work, you need to get a totipotent cell. And luckily, in many animals, every cell in the body, minus the sex cells, of course, because they only have half the genome, they're totipotent. They have the entire suite of genes. They have the entire genome. Now, in mammals and especially humans, it's a little bit more complex than that. You can't just take a skin cell and induce it to be totipotent because there are certain genes that have been shut down, converted into heterochromatin, so they aren't totipotent. But every nucleus has the complete suite of the genome, has a complete suite of genes, a complete genome. And so we can induce any cell, minus the sex cells, of course, to become totipotent by transforming the environment around the nucleus. And so what we do is we take the nucleus out of a cell and we put it into an egg cell. So the environment in the egg cell is an early environment, and so when you put the nucleus into there, it eliminates a lot of that the formation of the heterochromatin and allows the cell to once again function as a totipotent cell. And so this is how cloning works in animals, where you can't just take a cell and put it into the environment of an early embryo, because for many animals you can do that. You can do that with and you can do it with virtually every plant. But you can do that with a lot of animals. Vertebrates, it's a little bit complicated, but it does work for some vertebrates. But with mammals, you need to actually reprogram the environment within the cell. And so the easiest way to do that is just remove a nucleus and place it into an egg cell. There are some issues. So the chromosomes need to have sufficient telomeres. And so this is one of the things that complicates, just say you have somebody that's, 50 years old and you want to do therapeutic cloning or even reproductive cloning. And you take one of the nuclei out of, say, a skin cell and insert it into an egg that's been anucleated or enucleated, and allow it to become totipotent. The problem is that the telomeres, every time the cell in the skin, every time the not the germ cells, the stem cells divided to produce new skin cells, the telomere shortened. And so you're taking an old nucleus and putting it into a new cell, but the number of cell divisions possible is going to be determined by the length of those telomeres. Now, we can use telomerase to lengthen the telomeres. Telomerase, we talked about that earlier in the semester, and so we can use this protein to lengthen the telomeres and increase the longevity, and so now you'd have a totipotent cell that is capable of the number of cell divisions necessary to have a long-lived individual. And so what cloning does is it produces organisms with relatively identical genomes. And the reason I said relatively identical genomes is they're going to accumulate mutations that aren't exactly the same mutations that you accumulated. So if you're trying to clone somebody, again, from my previous example, that's in their 50s, you in your cells, or even in the early embryo, you accumulated mutations that were then propagated and passed down that aren't necessarily going to match now if you take one of your cells, you're 50 years old, you take one of those cells, it's accumulated mutations, but it may have accumulated mutations that aren't exactly like the mutations in your liver cells. and it may have accumulated mutations that aren't exactly like the mutations in the cells that give rise to your neurons. And then on top of that, this individual is going to accumulate mutations through cell divisions that you don't necessarily have um, in the rapid cell formation in the early embryonic stages. So it's relatively identical genomes, but the clone is going to have mutations that were not present in the donor, where the nucleus came from. But this can be originally cloning with Dolly the sheep was done to make a lot of copies of transgenic animals. And so this is still done with cattle and with sheep that were specially designed, are transgenic animals to produce human proteins, like copies of human growth hormone or copies of human insulin or copies of various human genes. And rather than waiting around for those transgenic animals to reproduce normally, you can clone them and produce dozens and dozens, even hundreds of uh, copies of those individuals without waiting around for them to reproduce normally. So that's originally why cloning was produced, and that is still a use of cloned animals. You can also use these to make copies of endangered species. There are zoos that are doing this, that are putting cells from various endangered species into freezers and are working to develop this cloning technology better so that we can use these to increase the reproductive rate of endangered species and try to save some of these species. So where there are some thoughts and some proposals with this in the area of human cloning is that this could be used to help people reproduce, those that can't reproduce normally, that can't that have some faulty mechanism. You could take a nucleus from one of their cells and help them have children that are a lot more genetically similar to them than a typical child. Also be used to help us study unique genotypes, and so there's been some thought and some proposals into trying to figure out if you have somebody that's incredibly unique, say Albert Einstein or somebody else, you could clone those individuals and try to figure out what some of those unique genotypes are. And then where we think a lot about this, because there aren't as many ethical concerns, is being used in therapeutic cloning. And so in here, let's say you have somebody that's suffering from cardiovascular disease, and you want to basically reprogram or get some early stem cells that haven't developed those same epigenetic factors that are in the current cells. And so what you would do is you would take skin cells from that individual, you'd grow them in the lab and get a lot of copies of them, and then you'd use the nuclei from those stem cells to clone that individual to produce an embryo, and then you could induce all of the cells from that embryo to actually develop into heart stem cells. And then you could implant those stem cells into the person's heart that they could then grow and divide and differentiate into the different cells within the heart and actually replace some of the cells that have developed some epigenetic factors that are contributing to the disease. And so this is an idea with therapeutic cloning. Um, there are ways you can do this using adult stem cells and reprogramming adult stem cells to behave like embryonic stem cells rather than using therapeutic cloning, and actually using stem cells probably has a lot more potential, but yeah, we can do therapeutic cloning. Now, some things you need to keep in mind about cloning is while they're relatively identical genomes, the gene expression is likely going to differ substantially. And so, which genes get shut down and how some of those epigenetic factors work change throughout the life of the individual depending on the specific environment. And so, while the genes are going to be virtually identical, the particular expression and which alleles are shut down and which regions of chromosomes are converted into heterochromatin. All of that may differ substantially, resulting in an individual that is incredibly different than the donor, even though they are virtually identical genetically. And then another thing you need to know about cloning is that it's extremely inefficient. And so you're talking on the 90% or more of the embryos formed when you take a donor nucleus and implant it into an egg cell do not survive. And obviously there are some ethical concerns there, because I think it's difficult to argue that just because you may have been the donor nucleus to put this into the egg cell, and it wasn't the normal progression of a sperm fertilizes an egg. I think you still have a hard argument against that. That's an embryo and that's a living human being. I mean, that's exactly how monozygotic twins work. Is you form a clone of the original embryo, and so I think you'd have a hard time arguing that embryo formed during that cloning procedure isn't an individual. And if a high percentage of those, 90% or more of those individuals die in that process, you have hundreds and thousands and even tens of thousands of individuals that are dying in that process, and that obviously carries some enormous ethical weight. And so, cloning isn't directly a type of gene therapy, but it certainly can be and has been proposed as a means of gene therapy with therapeutic cloning. And especially if when you take that donor nucleus and put it into an egg, if you modify the genome in some way and combine several of these technologies. And so, as we think through this and as we discuss it tomorrow, I want you to think especially about some of our ethical questions we need to ask and how we really think through this and work through this in a really intelligent and ethical way.